All right, guys, I'm here to take up your homework. So list 3 ways in which prokaryotes are important to humans and to the environment. So the first way that bacteria are important to the environment would be that they serve as decomposers. So they decompose everything out there. Okay, fungi, and you probably read fungi and bacteria compete to decompose. Bacteria are excellent decomposers. So in other words, they take waste products, including animal carcasses, and they break them down back into a usable form. So they recycle. They are important for food, so bacteria help us in the culturing of many of our different foods. For instance, they make sourdough. So when you want your bread to taste sour, you can actually throw certain bacteria in there, and their waste products, as they digest the sugar, they make a sour taste, okay? So that's one way you can make your food tangy. They are nitrogen fixers. So if you hear the term fixing, it means that we are taking something and making it into a usable form. So bacteria are able to take gaseous, so atmospheric nitrogen, and convert it into a solid fertilizer. And the fertilizer is used by the trees and the plants. And bacteria are photosynthesizers. So many of them are photosynthetic, which means that they make oxygen for us. Question 3 says, describe and state the function of each of the following. So a nucleoid is the genetic information. So it's the bacteria's DNA. So it serves the purpose of being the genetic information, okay? The pili are the extensions that come off of the bacteria. They help the bacteria to move, and they help the bacteria to share information with other bacteria through conjugation. A plasmid is a circular form of DNA. So this is like a chromosome, the nucleide, and the plasmid is that circle, and the circle is a form of DNA that bacteria can share with each other. All right, so when they are reproducing sexually, they're able to transfer their genetic information into another bacteria and receive the other bacteria's information as well. Peptidoglycan is a chemical found in many bacterial cell walls, and peptidoglycan it's an extra protective mechanism in the bacteria cell wall. So it serves as a protection for the bacteria cell, okay? So even though it can absorb antibiotics, the peptidoglycan actually protects the bacteria. A capsule is a slimy substance, a slimy chemical on the outside of the bacteria cell. Again, it serves the purpose of protection. It also serves the purpose of being sticky, and so the bacteria can stick to its food source or stick to another bacteria, okay, or to its host. So if it wants to infect something, it helps it to stick to it. And an endospore is a protective coating, lots of protection on these bacteria. A protective coating of the nucleus so that it can go, the bacteria cell can go into a dormancy stage. So in other words, it's not like hibernation, because in hibernation a bear doesn't shut down entirely, but in an endospore the bacteria actually shuts down. So it stops metabolizing, which means it stops using energy. And so it's in this state until it reaches the proper environment that it would like, and then it becomes active again. All right, 4 asks you to create labeled sketches of 3 common shapes of bacteria cells. So for that I have this. So coccus is round, okay? Bacillus, and it doesn't need to be, sorry, that could be capitalized and that could be capitalized as well. Bacillus is rod-shaped, okay? And spirillum would be spirals. And these guys are the hardest ones to find a real picture of, but that's basically what they look like. Just draw them like an S. Remember your prefixes. So if we have 2 coccus together, or coccus together, we have diplococcus, okay? If we have a chain, then we have streptococcus, okay? And if we have a cluster like grapes, and you would see this with bacillus and spirillum as well, then we have Staphylococcus. This is strepto, and this is diplo, okay? And once again, you can apply that to these guys as well. So just super quickly, if you had this, a cluster of bacillus, then you would have Staphylobacillus, okay? All right. Question number 5, distinguish between the following terms, inorganic and organic. Organic chemicals contain carbon compounds with the exception of carbon dioxide. Carbon dioxide is considered inorganic, okay? So you are organic, which means that you have molecules in your body that are strings of carbons, okay? That's organic. carbon-containing molecules with the exception of carbon dioxide. Obligate and facultative anaerobes. All right, inorganic means there's no carbon. Obligate and facultative anaerobes, when you see aerobes, you know that means it uses oxygen. Facultative means it doesn't have to use oxygen, but it can survive in oxygen. And obligate means it has to have oxygen. You and I are obligate anaerobes, okay? We're not bacteria cells, but we are obligate aerobes. Sorry, not anaerobes. We are obligate aerobes. So an obligate aerobe requires oxygen. A facultative aerobe can live with oxygen, it can use it, and it doesn't have to. Conjugation and transformation. Conjugation is when two different bacteria cells share their genetic information. So two different individuals will share their information. That is sexual reproduction. Transformation is when a bacteria cell comes across DNA and it's not in a living form. It's just finding DNA somewhere and it takes it into its plasmid. So this is going into the plasmid with conjugation. The DNA is going into the plasmid as well with transformation. But with transformation, the bacteria is not sharing it with another living cell. It's just coming upon it. So usually it can come upon bacteria DNA very easily because the bacteria are dying quickly. And the other thing is it can come upon another species of DNA as well. And sometimes it can actually take on other species' DNA. So that's what makes bacteria so fascinating, the fact that they can just pick up DNA and incorporate it into their plasmid. Question 8. What is the benefit to one kind of bacteria producing antibiotics that kill other types of bacteria? Well, it decreases the competition. So if you are an antibiotic resistant bacteria and the bacteria around you are not antibiotic resistant, so penicillin kills them, you now have more resources, okay? So you can use the resources and you have less competition, just like in evolution. Describe the process by which many bacteria have developed resistance to antibiotics. So the number one way that any organism is going to evolve is simply through mutation. So as bacteria reproduce their chromosome or chromosomes, if they make a mistake and that mistake is beneficial, it allows them to be antibiotic resistant, that's how that comes to be. Okay, so it's just because they reproduce so quickly, they can make many mistakes in a very short period of time, and those mistakes can be harmful, they could be beneficial, or they could be neutral. So we're talking about a beneficial mutation here, and that's how bacteria can become resistant to antibiotics. They can be living in an antibiotic-rich environment. So in other words, you and I, if we take antibiotics, and one or two of them can mutate to be able to survive through that antibiotic. How has their ability to reproduce rapidly influenced this process? If you take penicillin over the course of 7 days, those bacteria are being exposed to that penicillin for 7 days. One or two of them can mutate, okay? And they can mutate to form an antibiotic resistance because they are reproducing so quickly. I showed you that diagram. You can make 2 bacteria after 20 minutes. You can make 4 bacteria can make 4 bacteria after 40 minutes, and it just goes on and on. So because each bacteria cell has the ability to reproduce so quickly, they can adapt to their circumstances because they can mutate as well, okay, and mutate quickly. Describe 2 examples of symbiosis involving bacteria. So hopefully you looked that up. I'd love to hear your examples, but I can't. So I will give you 2 examples. You can have bacteria that live on roots of trees or other plants, and the bacteria gets some food from the plant, for instance, whatever the plant is making, and the bacteria provides the tree or the plant with nitrogen because it's fixing nitrogen into a fertilizer, which is awesome for the plant. Another example is bacteria living in the termites. So in living in a termite digests wood. So the bacteria allow the termite to digest the wood. It's the bacteria that are actually breaking down the cellulose in the termite's gut. So imagine a termite really does have a tiny little stomach. The bacteria in that stomach or that gut is able to transform cellulose into something that gives the termite energy. In addition to that, you know that you and I have, and we'll go over this again in the digestive unit, we have E. coli bacteria living in us, many different types of bacteria living in our intestine. You can get those in probiotics, you can get those in yogurt, and they're there to help us digest our food more rapidly so that we can get energy from it, okay? And get rid of our waste products even more rapidly. All right, have a great weekend.